Welcome everyone in today's second day webinar related to QSAR, machine learning, AI for the drug discovery purpose. Welcome everyone. Uh, let's hand over to our resource person, Mr. Shankar Thapa from US. Shankar sir, you can proceed ahead.
Mhm. Hi, good evening, everyone. So thank you, Dirgha sir. Yeah, so yesterday we learned a little bit about the QSAR and its like theoretical approach as well as we try to learn one software from where we uh, design a model on normal database. So that software is good. That software use the machine learning approach and AI to design models and validations of it. That software is pretty new software. I think 2 years ago or 1 years ago only they have developed. So it's not that much used in publication things. So you can use, but like the use case of that software is not huge compared to today's software. So and then like another drawback, whatever this yesterday's software, so RAN QSR software was, so the problem with the overfitting is there quite, because like we are using the machine learning thing in machine learning QSR model development. Overfitting problems is a big challenge. So to overcome these types of things, okay, so obviously overfitting will be there in every software. So yesterday's software didn't allow us to understand what goes behind the model development, right? So how we can prepare the data set, how we can clean the data sets, those all part was inbuilt in that software. So today uh, we are going to learn another software which is
Shankar sir, are you still talking? We could not hear you.
yes sir. Yes, sir. Yes, sir. I think like I
Okay, okay.
blocked my this mic. So okay, so that's it. So today we will learn about another software, and this is pretty much good for the publication purpose because we can get all the required plot from model development and then model development equations. So and yesterday software that ran QSR software, in that one I think it's quite difficult to get the equations, so because that is AI based or the ML based. So in today's software, the software name is QSAR. So the software is developed by the European scientist Grammatica. So Paul, Dr. Professor Paul Grammatica. So I think he's from France or somewhere else. So and then software is freely available. So, but another drawback of this software is we can't uh, download it from the website. So we have to request, basically your PI should request for the software. Okay, so we have to mail the professor and then the professor will send you the software files and all the required documents. So in the mail, you have to provide your previous experience and your ongoing project. So why you need this software? So in that case, the professor will send you mail directly. And another criteria to get this software is the mail ID which you are using to mail the professor should be academic mail ID. So from personal mail ID like the Gmail ID. So uh, he is not going to send in that case. So those are the case. Okay, you try to get the software in that way. If you are not getting, so I have the access and then I can share with you. So why I don't want to share right now is because it's quite unethical. So I have to take the permission. So because like some developer has done that much of hard work to develop the software, so it's better to get from him. Okay, so in any case you are unable to get the software, so I will provide. Yeah. So with that, so I'm going to share my screen. I hope my sound is audible to everyone. So can anyone confirm it?
Yes, sir.
Okay, so thank you. So here is my screen. Hope it's visible to everyone. And then, yeah. So the first things is my screen is visible.
Yes, sir, it's perfectly fine.
Okay, so let me take you to the software website. Okay, first of all, so you can type QSAR software. Okay, in Google. So from there, like you can uh, go to this website. This in Subiria is there. So this website like QSAR software for MLR model development. So this is yesterday we talk about multilinear regression approach. So and then this is genetic algorithm based MLR model development we can do. So you just click here. So this is the way how you can access the software. So this research group has developed so many other software, including this one. So including they also developed the software from where you can predict the toxicity and all the things. And QSAR classifications approach also they have developed so many things. So you can go there, and then if you click to the software option here, so you can get the access of all of these software. So these all softwares are freely available software. So you can download all of them and then use it so for your learning purpose. Okay, so the required software is this last software. Okay, last option, the QSARN software for MLR model development and validations. This second last is also good, but this is we can't develop the equation type of things in this one. You can basically use it for the like toxicity types of things, right? So, but this is the software which is required to build the QSR, and this is widely used in academic research. This software is like, and then like uh, thousands of publications have been already uh, conducted using this software. So you click here, okay? So yeah, after clicking, you will be directed to this page. Okay, so QSRN software for the this development. So here they have clearly mentioned to obtain a copy of QSRN, please read the following instruction document. So this document is there. In this document, they have provided the email ID also, and then the instructions also. So the software is developed by Professor Paolo Grammatica. So from I think yeah, he's from Italy. So they have developed. Okay, so to get it, so you have to mail to the professors. So this is the mail ID. So here they have so clearly mentions QSRN can be obtained by sending a request to Professor Paolo Grammatica, illustrating the personal experience in QSR modeling. Basically, it is suggested that your guide or your PI should mail, and then like chances of getting mail reply from the professor is high in that way. And then please briefly explain the personal experience and for what reasons you require this software. So that is also very important. So other than that, so this is like a tutorial type of things. This is tutorials. Okay, so you can go through it. So in that way you can obtain the software. So professor will mail you, and then in mail he will attach the required paper. And if you are using the software, you have to cite those paper. Okay, and then as well as the software file you can get it. So this is the way how we can get the QSAR software. It's free of cost. Okay, so only the thing is you have to mail. That's it. Okay, so I think this is clear. So now I'm going to yesterday. If anyone remember the procedure, okay, just one more time I'm going to show you the procedure. That's it. So because we are going to follow each procedure. Okay, so in today's session, so all the procedure we are going to follow. So the first steps was here, right? So it's a data curation. So chemical data we have to make. So yesterday also I briefly explained about how we can extract the data from available resources, or another approach also I explain, like from the published research paper. Okay, so only one research paper also you can take, or the multiple research papers which are related to the same cell line activity, and then same unit of IC50 value is there, so you can use. So this every steps we are going to follow one by one in today's sessions, because it's required and it will give you the comprehensive idea, or the confidence to develop the models. Yeah, so these are the things we are going to follow today. Yes, so you can take a screenshot for your convenience if you want. Yeah. So first things, we need data, or we need the compounds. Compound in a sense, we need their structure in SDF format in 3D. Okay. And another one is their biological activity, which means IC50 value in micromolar unit. And then the cell line should be same for all the compound. So yesterday I showed you how we can do from one server, right? So for that one, we have to go to uh, Campbell ID, right? Yes, Campbell database. Yes, yeah, yeah. This database. Okay. Today we will try. Okay, can anyone uh, type their research on which topic they are doing, so that I can try to find out the compound related to that particular things. Can you type, anyone, please, so that I can do for your convenient in that way. Of course, it is going to take a time. That's why, like, I have already built database, so I will use those one for the demonstrations. But I will show here once again how we can do. Please tell me any activity on which you want to do. Is there any? No.
Someone typed colon carcinoma.
Yeah, so in colon carcinoma, also particularly like which activity you want to do, or like which particular pathway you want to break. So if you are going in that depth, so it will be easy to get accurate data. So don't go surface levels, like in terms of like diabetes also, somebody has written their diabetes. So in diabetes also, like which pathway you want to particularly target, and then like uh, which activity you are interested in getting. So like in our underdeveloped country, most of the people follow like alpha amylase inhibition, alpha glucosidase inhibition. So in that sense, like we have to find out in that way. Okay, so let's say the colon carcinoma. If you do in this way, like you will get huge data, so it will not be specific to your study. So here like 9.8K assays are related to that colon carcinoma. So they have used the different cell line, right? So on which cell line you are interested, so that cell line study you can type. In that way you will get your specific data. Okay, so that is one reason, because uh, curations of accurate data and then authentications of data is very, very important, and then crucial steps to develop the accurate QSAR models. Okay, so that's why like it's very important to do that. So let's say this ACT8 colon cell carcinoma cell lines is there. So okay, so it's cytotoxicity basically, and then organisms is Homo sapiens. Okay, so you can do, and you can use, if you already know from the paper, if you know the this ID. Okay, Campbell ID, if you already know, so you can search by this ID also. Okay, so let's click this one, how many compounds they have used in this particular experiment. So on the basis of that, we will be able to extract the data. So after that, yesterday I told just go to the activity chart. Okay. So here IC50 they have given. So please remember, sometimes like they can give multiple this activity, this ones. Okay. So sometimes they will give IC50 value, sometime enzyme inhibition, sometime KI. Okay. So suggestions for the QSR model development is either you have to go for the IC50 value or KI, so which is a binding affinity. Okay, so experimental binding affinity. I'm not talking about the docking, that part. So I'm talking about actual experimental binding affinity, KI. So that two things are like mostly used in QSR model development. So okay, so sometimes you can get the multiple activity chart over here. Okay, so that time also you have to be specific. Either you need the IC50 or KI or some other parameters. Okay, so in this one only one activity they have performed. So they perform on total of 28 compounds, right? So it's quite good. So 28 compounds is fine for QSR developments. Okay, so you just, after that what I told you, just click here. Okay, yeah, once you click you will be directed to this page where you can get all the informations which required for us. Okay, so like we need the standard value, that IC50 value, so which is given here. Okay, so in this way IC50 value is given, and then compound structure is also given. Okay, so this way, and then if you drag this right side, okay, unit is also given, as well as what type of activity it is. This is cell based activity. Okay, sometimes you have to verify these things also. Okay, and then this cell line also, just make sure that all the compound use the same cell line, and then same cell based assay, and then same unit. Okay, so here also you have to check this unit, this nanomolars is there. Okay, and then same organisms, on which organisms they perform the activity. So in this case mostly they perform on same environment. Okay, we can say. So yeah, so cytotoxicity, cell line is same, so everything is same. So these all parameters you have to check before downloading or using those data. Yeah, so I think it's pretty much clear that every compounds have the similarity in the environment. So you can download it. So how you can download? You can click here. So it will select all the 28 items. So here it is written that 28 items are selected out of 28. Okay, so now you have to download here CSV file, or I would suggest you TSV file if you download. So if you are downloading CSV file, it's quite difficult to get the desired IC50 value. So please make sure that, so these steps we are only doing to get the IC50 value. Okay, so this is only for IC50 value. So this first downloading TSV file is only to get the IC50 value. That's it. Okay, so for structure we are going in next step. So with yesterday also I explained. Okay, so that I will show you just in second. So it's just download it here like TSV file format. Okay, so it's generating. Okay, so it's 100%. It's already generated. So yeah, so now the download options you can get in this side. Okay, so download the file. Yes, so this is pretty clear that we got the file here. Okay, so let me go through the file. So it is in download option. So this is in zip file. So you have to extract it. Okay, so extract, done. So let me delete this zip file. So let me give it the name. So this is only for IC50. So let me give it the name IC50. Okay, so you just open inside this. So you can have this uh, file. Okay, TSV file. So now how to open this TSV file? If you are going to open in notepad, all the informations will be in like another format, so you can't extract the information. So the better way to get the informations, desired informations, is, so you just open the Excel. Okay, Yes, so you just open the Excel file, and then after that you open that file. Okay, go to open, browse, and then download IC50. So here, like, because I have only selected the Excel file, let me select all. So yeah, now it's visible, right? So you just open it. Yeah. So you will be directed to this page. So this is information. So this should be checked, okay, delimited, and then next, this should be checked tab. So if you see here this preview, okay, so columns, separate columns are there so that it will be easy for us to get the informations, and then click next, okay, and then again click finish. So now look at this. So the desired informations is in column format so that like it's easy for us to get the IC50 value. So here the standard unit is their IC50 value. Okay. So the value is given here in this line. So this is the IC50 value. Okay. So all of the compounds following the same unit, and then they have the exact IC50 value. Okay. So everything is there. So we only need, I already told, we only need this IC50 value. So remaining things we don't need as of now. Ismail formula, you can keep it. Okay. So because that is very important. So remaining columns, you can delete it. Okay. So yeah, this molecular ID also you can keep. So remaining, you can delete it. So you can left click and then drag in this way. So you can delete it. So right click here and delete it. Okay. So unit we already know because we are working on nanomolar unit, so it's not required. Just delete it, and then remaining things also you can delete from here. This is how we can make a file. So yesterday, whatever the file I have used for that software, so I prepare that file in this way only. Okay, so let me write it this IC50 value. I think I'm clear in this. So if any confusion is there, please let me know so that I will try to resolve that. And then please don't forget to save it. Okay, so you have to save it. So go to file, save it as. Okay, so let me save where I want to save. Let me save in webinar. Okay, so let's say WAPINR webinar IC50. Okay, so you check it should be in TSV file format. Okay, so like CSV file format is fine. That's comma delimited CSV. Okay, yeah, save it. Done. So now please remember this is IC50 value. Okay, very important and then crucial point to remember. So this is IC50 value, but we require it in form PIC50. Okay, so PIC50 is nothing but it's a minus logarithm of IC50 value. Okay, so if you do in that way manually, that is also fine, but I often suggest everyone to use the server. Okay, one server is there. So we understood now, right? So we got this IC50 value from database, but to curate the QSAR models, we need PIC50 value. So this value need to be converted into PIC50. So for that reasons, either you can use manually also by using the formula minus logarithm of IC50, okay, or something is there. So something like that formula is there. So you can use that, or the best way, the error margin will be very less if you are using the online server. So for that one, you just go to the online. So type here CONVERT. There's a Hunter. There is a website called Hunter or Hunter Convert. In that one, you can do IC50 to PIC50. So yeah, so this is the Drug Hunter website is there. Okay. So yes, so here you have to choose your accurate unit. Okay, so our input is IC50 value. So let's say for first compounds, what is 500, right? So let me type 500 here. Okay, so now our PIC50 value is 6.301. So you uh, like this, type it. So accordingly, for all the remaining compound also you can do. So this is 1540, right? So in this way, so that is 5.812. Uh, I think this is clear. So accordingly, you will do for remaining compound also till here. Okay, so that will be our required data. Am I clear? Is there any confusion? So that if no confusion means I will move to the next steps. So let me remove it. I think I'm clear. Okay. So please be interactive little bits. Otherwise, like, so I'm not getting any informations either you peoples are listening or not. Yes, so this is for IC50 value. So we did it to get the IC50 value. Okay, so we selected 28 compounds, we choose screen out and everything. So we got the IC50 value and we converted IC50 into PIC50, which is required. So now, second things, we need the structure. Okay, so structure in 3D format. Okay, so preferably like SDF format in 3D. Okay, so for that one, yesterday also I explained this step. So you have to click here related compounds. Okay, so options is there. If you click the related compounds, it will direct to these 28 compounds in SDF format. Okay, so that we can download it. So you just click to related compound. Okay, so now it is clearly showing 28 items compounds. Okay, so if you go down, scroll down, so yeah, so total 28 compounds are there. Look at this. Okay, so in another way, if you want to visualize this, you just click here. Okay, so it will show in column. Yeah, the same way, if you click it, it will show in row format. So any format is fine. Okay, so if you select all here, so it will show again here. Okay, selected 28 out of 28 items. So we selected all the 28 items. So now if you go here, we have one more options. Okay, before these options was not there. So now it's one options, SDF options is available. So which means this is structure format is okay. So this is for structure. So you just click it. So again here they are generating the SDF file format. Okay. Yes. Yes. So generated. Now I can download it. This is downloaded. Okay. So again, let me go to uh, download section. Yes. So this will be in compressed file format, so zip format. So I have to extract it. So click extract. So and any way you can use. Okay. So if you have a software to extract this, you can use that one also. So no problem. So let me rename it so that it will be easy to remember SDF. Okay, good. So open it. So let me rename this also SDF. So now you should have a software to open these things. Okay, so yesterday I suggested some of the software. Either you should have CameDraw, or you should have this a software called Marvin Sketch or Marvin View, or preferably Discovery Studio. Okay, so any of the softwares is uh, okay to open these things. Okay, so now SDF file formats is there. So all the 28 compounds are in this one file format. Okay, so if you want to check, so you just right click. So in Notepad also you can check, so that you will get the idea regarding the dimensions of each compound here. Okay, like this. Okay, so now, or if you want to open with any software, so I have this BioVia Discovery Studio. This is also free software you can download. Okay, so let me open it through it. If you want to extract all the file into individual file, that is also fine. Okay, so if you don't want to extract, you want to use the same file, that is also okay. So if you want to extract individual, so you click here. Okay, so this is one. So now you have to save it to file, save. So individually you can save it into PDB format. Later you can send PDF format. So this is one compound. So So you just click this one, okay, molecules 2, second compound, molecule 3, and this one you can visualize these are the molecules, okay. So these are the molecules downloaded. Now all these 28 molecules are in one single file, which is fine. Okay, so no need to, it's not compulsory to separate file you can use. I think now I'm clear. So any questions is there? So let me okay. Yeah. So now we have three structure in SDR file. Part is, I think it's not mentioned here. Previously, I have mentioned some energy minimizations. Okay, so this is important. And then one more thing, uh, very so understand here is curation. We have to be very careful regarding the protonation state of the compound. Okay. And then salt conditions, tautomerisms. These are the some points which we need to be very careful. Okay. That reasons for that to check one, so you can download, or sorry, you can check individual file by opening it in the software. So what is the protonation state? What is the PS?
Shankar sir, are you there? I feel some sound disturbance and now looks like he's disconnected. Let's wait. Shankar sir, are you hearing us?
Yes, yeah. I don't know what's problem is there. So
Now it looks fine, sir. Yeah.
okay. So till now, I think it's clear. If any questions is there, I will be happy. Otherwise, like I will move forward.
So your screen is not visible.
Yeah, now like I can do this one. Okay, so that's fine. So I was saying I was giving some information, so which requires to validate. So that was like I was telling like, the structures we have to be very careful. It's like either the structure is accurate or not. Okay, of course we have downloaded these things from the verified database. Okay, so Campbell's is it's authentic and then verified database, but still cross verifications is very important. Cross verifications for protonation state, cross verifications for salt conditions, cross verifications for tautomers, or cross verifications for ps, and all the things it is important to cross verify. Okay, so that you can do by opening individuals compound in software. Okay, so that is the reasons. Yes, so let me again share. Now we are going to do the energy minimizations. Okay, so yeah, so energy minimizations we can use. So this it should be, yeah, I think Zenith the essence is raising the answer. Yeah, please go ahead if any questions is there.
Sir, you're taking this IC50 as kind of a standard parameter. Is there any other parameter that we can choose?
Yeah, ki value you can choose, as I previously mentioned. Yeah, which is binding constant, what we call like that is the binding affinity of drug with the protein. So experimental ki value you can take.
So
Other
for
than
that,
that,
do we have to first go for docking first?
no. I'm talking about docking affinity. Okay, docking is computational things, right? So in QSR development, we don't consider the computational value. We need experimental value. So I'm talking experimental binding affinity.
Okay, got it. Is there
Yeah.
any other parameter? Suppose can we take bioavailability as one of the parameter?
Yes, so if you are plan, it depends on your research projects. Okay, if you are clearly defining the problems or the objectives, you can use that one, but that will be only applicable to find out the relations of your bioavailability with the chemical structures, right? But here we are seeking to find the relation between your descriptors and activity, biological activity, right? So that's why you are using IC50 value or KI, which are the standard to develop the QSR models. Suppose you are designing the QSR models to define the toxicity or to define the bioavailability. Okay, so which you are talking, you can use, but the use case of that models is limited to particular that area. Yeah.
Okay, thank you. You can continue, please.
Okay, so now energy minimization. So why we need to minimize the energy? Because like in the computational things, what we believe is the energy state of the compound should be in local minima. Actually, there is a theoretical part like global minima and local minima. So in that way, like so the if energy of the compound is optimums or the low or the so that compound is considered biologically bioactive but not biologically active, so that compound is considered as a bioactive compound. So energy minimizations and geometry minimizations, for that reasons we need to minimize the energy. Now there are different force field for the energy minimizations. Okay, so many different force fields are there. UFF is there, one of them. Okay, and MMF 94 is there, MM2 is there. Okay, so other recently developed new force fields are also there. And then very important another one is there, DFT, density functional theory, which takes little time, but you can use that one for the energy minimizations. Okay, so these are the parameters you can use. So how you can do one things you can do by using Open Bibles. Okay, Open Bibles is also free software. This one you can use Open Bibles for the energy minimizations, but problem in this Open Bible is manually you can't do energy minimization through this software. So what you need the Ubuntu systems. Okay, so through Ubuntu system you can minimize the energy. Okay, so that is one drawback with Open Bibles. Or another way of minimizing minimizations is you can use the RD kit. Okay, so let me just show you because I have developed one uh, prompt for this one. So for that one, I will use Google Colab. Okay. And before I was talking about the cross validations of structure, right? So this small command prompt I have prepared. So if you are using this command prompt, okay, so whatever the protonation state and whatever the hydrogen's ion concentrations or whatever the polar ions or tautomerism form will be solved. But like as of now, I have only developed by individual compounds, like developed to individual compounds. So only one by one you can do. So I'm planning to add some more code here so that we can use for multiple compound also. So I will share these things, so I can share this to you in the group so that you can use this for the energy minimization. So here I have used MMF force field for energy minimizations. Okay, so you can do so. There is no difficult step in this one. You just have to click each and every steps here. So you just have to click this because I am like installing the RD kit first. Okay, so first you have to connect this. Here it is connecting. Okay, so then after that you have to, these all the informations I am giving you to make you what we can say, confident developing the software. Because the if you know the different way to do one particular task, so it will be easy. If one way is not working, you can shift to another way. uh, One server is there which we are going to use, so that server itself do the energy minimizations also, so you can directly move to that one also. Okay, so I will show you. So you just click this. So these are the dependency which required to do, and then you just click it here. So this is the step where you have to copy your smile formula of your required compounds and then paste it, okay, inside this double inverted comma, and then you just hit here, you can get it. Okay, so you can get this compound, and after that you just click these all the things, and then by the end of this you will get the structure here. Here you have to go. So here you will get the structure. That structure you can download. This is another way to for the energy minimizations. Okay, so yeah, one open bible, but open bible one limitation I told you, so you should have a ubuntu system. Okay, so another approach I have given this link. Okay, I will keep this link, I was saying, so that you can use. So let me share in the group chat. Okay, so you can keep it with you. Yeah, so this is second steps. Okay, individually, if later I will explain, like if you need how to do the energy minimizations in Ubuntu system, so that I will do, because it's lengthy process. Again, I have to go open the Ubuntu and everything's okay, and one code is there, we have to keep that one. That's why I'm not going to do that one. Yeah, so this is energy minimizations. That is also done. So now after energy minimizations, we have to calculate the descriptors. Okay, so now this is the last steps for the data cleaning, or the data that compound that one. So descriptor we need, right? So yesterday also I have given some of the software name. So one was Paddle. So Paddle you can download, or very easy, Chemdes. So if you go to this website Chemdes, okay, you click it here. So this is the website link. I think yesterday Dr. Dirgha sir also share this in the group. So again today also I am going to share. You can use it. Okay. So this link. Okay. Yes, so this is the user interface for this Chemlist software. Either you can log in, or you can use as a guest also, no problem in that. Okay, so now how many molecular descriptors available in this server? So look at this. So first descriptor is ChemPy descriptor. So how many descriptors are there? 1135. So CDK descriptors 275, RD kit descriptors 196, Table descriptors 24, Blue Dex descriptor 174, and Paddle descriptors 1874. So mostly we are using the Paddle descriptors and RD kit descriptors. These two descriptors you can use, but we can go with the Paddle descriptors. Okay, so 1875 total descriptors it can calculate. So how we can do that one? You just go to here web server. Okay, so here that the same options you will get it. Okay, so now you just click Paddle descriptors. Okay, yes, it's that easy. So now either you can input the individual compounds smile formula here, and then seek these options. Okay, so either you want to, which descriptor set you need, 1D and 2D descriptors, or 3D descriptors. Okay, so or all, like 1D, 2D, and 3D also. In that way also you can do. And here is one option available. First option is convert to 3D. Understood? So if your compound is not 3D in structure, you can convert. And another options is there, like energy minimizations by using this two force field. So no need to do energy minimization separately if you are using these web servers, right? So you can check this one. Yes, I need to minimize the energy, and then you can upload the individuals file format here, or the smile notations, so that you can submit. This is one way. Okay, and this is lengthy way because for individual compound you have to do. Then what is the shortcut? So here now you can download that SDF file format here, okay, for all the 28 compounds, and then you can select your desired descriptors 1D and 2D or 3D. Okay, so and then you can choose these options. So if you want to use the MM2 force field, or if you want to use the MMF94 force field. So, but I suggest you use MMF94 force fields. Okay, so please you can study about the force fields. What does it means? I think in previous webinar I have explained a little bit about this force field in during documentations and all the things. Okay, so this is the way. So let me show you how to do. You just choose the file format. So where was the file format? This STF, right? So this STF file format. So my file format is uploaded here. So now I will choose the 1D and 2D descriptors. Yes, I want to minimize the energy. Okay. So now submit. Sometimes this web server doesn't work because yeah, we all know this is free open access. So that so many server load will be there, but like most of the time it does work. Okay, so that you can get our descriptor. How many descriptor we are going to get? Around 1875 descriptors we are going to get. So it takes time, okay, generally, uh, because we are converting into 3D, and then as well as we are minimizing the energy, so it will take time on the basis of your number of compounds. Okay, sometime it may takes 1 hour, sometime even 2 hours, or you have more compounds, it may takes 3 hours. Okay, so yeah, so this is the way to do. Okay, so let me show you. Yeah, so you can get at the end the result here in CSV format. So that Excel file format you can download, and then so that Excel file format you can download it. So that Excel file format will be something like this. Okay, let me show you. Something like this. Yes, something like this Excel formats, or around 1800 descriptors will be there. So these are the descriptors I have screen out. So like this file format you will get. So yeah, pin descriptors. Okay, so let me show you. So these types of descriptor you will get thousands. So these are like few. Here few descriptors are there, but you will get around 1800 descriptors. I have deleted the unwanted descriptor. So now in this step, you have to be very, very careful. Now in that previous file, we have a PIC50 value, right? So you have to assign the accurate PIC50 value of your compound. So you have to take the compound name. So accordingly, you have to assign the PIC50 value in this one. Here I have assigned in the end. So here I have given that PIC50 value. So this you have to be assigned very carefully. Otherwise, if you mismatch the value, then your QSAR model is gone. Okay, so we have this plenty of data now. Okay, so now we have to follow the ORCID ORCD guideline also, and then all other parameters we have to follow. I think till now it's clear, right? So I think you must have some queries. Okay, this is so lengthy. Okay, so many things we are doing. Yesterday's server, that software was easy. In that one, no need to do anything. Okay, yeah, that's true. But if you use that previous software, you will be not knowing what goes into the development of QSAR study. Okay, so hopefully till now it's clear. So now we are about to reach to develop the models. Okay, any questions?
No, sir, actually, these are all the step by step you're showing, so no questions.
Okay, so now this is if no questions. So now I will show you how to screen out or how to find out the desired descriptors. So look at here. So an acid, log P. So these are the descriptors, right? So these are the descriptor we never seen in our life, correct? So during QSR model development, we will get to know these types of descriptors are also available. Okay. So now unwanted descriptor we have to remove or delete out. So what are the unwanted descriptors? If your value is 0, remove it. Please note down, or like be mindful, because these are the very important steps. Okay, so if you do any mistakes, your QSR model is going to, crash, or you can't get the good models. Another one, if your value is similar. Okay, so around like 80% of the values are similar, like 18, 18, 18s are there. So if this value, 80% value are similar, means you can delete it. So I already have told 0, you can delete, correct? Another one similar value also is there, that descriptor you can delete. That's what I mean. Okay, so these all 0, 0, 0, 2 similar descriptors. So in this way you can delete. So actually this is the lengthy stage, so it takes 1 hour to clean your data accordingly. Okay, so two criteria I told. One, similar value more than 80% is available, when you can delete. Or another one, if value is 0, that you can delete. And another one, if your value is unit, means 1. If your value is like 111 is there, right? So that is all, that also you have to delete. Okay, so this all the value you can delete. So similar is there, so this one value, two value, so in this way. So accordingly you can do, because now very few descriptors are available. But you just remember that you are screening on 1800 descriptors manually. Okay, so that you have to do, because it's a learning, so that you will better understand the QSR model development. So accordingly you can delete. Okay, right click, delete it. So accordingly you can delete the unwanted. So at the end, whatever the value left by cleaning these all data, all the unwanted descriptors, you can use those descriptors to develop the QSR models. Now we are done with data management and data cleaning and all the things. I think I am clear.
Sir, sorry to interrupt. One question. So,
Yeah.
is there any software which can do this job for us, cleaning, data
No.
cleaning?
As of like my informations, particularly software is not there, but if you are familiar with the Python language, so you can do this thing in Python
And
or by
also,
using the
like,
Colab.
also if we go manually, so by
Mhm.
mistake, if we like delete some main data, one or two
Mhm.
columns, so will that affect?
Mhm. Yeah, definitely that will affect. That's why, like, while cleaning this data, we have to be mindful. Yeah.
Okay, thank you.
Okay, so that is the thing. So, okay, it's not that much difficulty. So it looks difficult, but it's not that much. Yeah, of course, we have a software today. We are using that QSAR software. That software also does this part. Okay, most of the part, but not all the part. Most of the part that software also does. But before going to that software, I suggest everyone go through manually, and then whatever the remaining things is there, so that software will take care. Okay, so am I clear? So now I can go to the software, main software development. So the now you must have been understood that the data collections, data cleaning, okay, and their validations is big and very crucial steps in QSR model development. Either your models will be successful or not, depends on your data cleaning skills, data, validating capability. So now I will open the QSR software. And please remember, we have assign the PIC50 value in the same file where descriptors are available. That also keep in your mind. Okay, so don't forget that steps. I told you. So assign the PIC50 value, accurate PIC50 value to the same descriptor files in one column. So I'm going to use the prepared data set here. So this is the what user face of QSAR in software. Okay. So one good thing is you have that paddle software incorporated in this one. So if you want to do these steps, which we are doing, yeah, so now you got it right. So here you got the descriptors. Look at this. Okay. So this many descriptors you are getting. So altogether 1875 descriptors. Okay. So now you can download it. Yeah, actually it doesn't take that much of time, right? So it takes hardly 10 minutes. Yes, so these descriptors, and after that I already explained. So in this way you can use this ChemDAS server to calculate the molecular descriptors and energy minimizations. The same process you can use by using the actual software. Okay, so here in QSRN that is also available. Run paddle is there. So if you click this one, you will be directed to the software, paddle software. So this is the paddle software. Okay, so this is for the descriptor calculations. We use the same paddle software information, both things here, because we use the paddle descriptor, right? So both things are same. Either you can use this software or you can go with the server. So this is also good. So you can input file here. Okay, and you can choose the same thing, 1D you need or 2D you need or both you need. Okay, so here I was talking about the that important points like salt conditions, okay, aromaticity, tautomers, and those all the things you can check here as per your requirement. And then other parameter also you can say 3D parameters, fingerprint parameters. Okay, so at the end you can run start. So this will take time, and then but you can do the same thing here also. Yeah. Okay, so now if your data is ready, now you have to upload that data here in QSRN software. So file, go to file. Okay, import data, go to file, import data. Now I will go here. Let's say, let me, because I have already screen out or clean the data, so that let me use this one, paddle descriptor. So this is clean data, or I don't know. Okay, open it. So after clicking that, you will get these things. Okay, first steps here, delimiter is there. So in that one you have to write comma. Okay, if you are writing comma, if you are giving comma here, so that it will be directed to all the descriptors of 71 compounds. So I use the 71 compounds. This is my old data. Okay, and then like all the descriptors available here, right? So 886 descriptors are there, correct? So after that, so that I think you are asking, is there any software available to do cleaning the things? Yeah, this is available here. Some of the cleaning can be done. So here you have an option, exclude if constant is greater than 80%, which I explained, like manually also you can do. But in by chance, if anything is missing through manual way, so you can do here also. So you just click it. So, and yes, okay, yeah. So now this is going to be screen out that one. And here exclude if your correlations is above 95%. So means the any two descriptors are very close to each other, means it will exclude that one also. So you check that one. Yes. So now after that you have to hit enter, import here. Here option is there, right? So import. Okay, so now our data is imported, and then here we can get the informations about our descriptors. Okay, how many descriptors have been selected after cleaning that 80% and 90%. So here you can see 485 total excluded descriptors. So this many descriptors are excluded out of 800. Okay. From 1800, I did manual screening. So I reached to 800, and then from 800 also around 400 excluded. Now you can consider maybe the 400 descriptors are there, which we are going to develop the model on those descriptors. Okay, so first steps we completed. This is data import steps. So second steps is here, go to analysis. So you can see those the data again. So if you click the view data, so it will show the same data. Okay, so now different here visualizations pattern is there. So okay, so you can click and check. So these are the data. So visualization pattern. Okay, so here you can choose on what basis you want to visualize. Okay, so this is not that important. Yeah, but still you can visualize the things. Okay, so yeah, and then you can go to second steps, data setup. So now we have to set up the data. So click data setup. So you will be directed to this panels. Okay, so here one side is variables. Okay, so another side is data set, what you can say data sets. Okay, so now all the data are under training status. Okay, so training data set you can say, but in theory yesterday I told we have to divide the data or descriptor into training and test, right? So in the ratio of, like I told, like 30 is to 70 is the standard one, like most of the people use that one. But if you are not getting the better QSAR models through 13 to 70, you can change that parameters, 75-25 or 60-40 or 80-20. In that way you can divide the sampling. Okay, so the first steps is we have to assign which is variables and which is response. Remember our IC50 value or PIC50 value is response, that is our response. Okay, so other parameters or descriptors are our variables. Okay, so how we have to select that? Just left click in your mouse and drag down this all the variable except your IC50 value row. So this first row is IC50 value. So if you have given the IC50 value in first row, it will show in first row. If you have given in the last row, it will show the last. Okay, so now I have given in first row means. So now accept this IC50 value, select the variable. So all the descriptors are variables. So left click in your mouse and just drag down. So manually. So why we are doing is we are like dividing the data set into training and test. For that reason, we have to define, okay, which of the data is variables and what is your response. So it's like more about a dependent and independent variable. Yes, done. So after doing this, you just select here, all the options is there, select variables. Okay, done. So now this is selected. Okay, so now second steps I told you, response. So which is our IC50 value. Just first select it and then you select response. Options is there, select response. So now our IC50 is response and other remaining descriptors are our variables. Okay, so here everything is selected now. Okay, so now we didn't divide the data set. So for dividing the data set, we have an option here. Okay, so like random percentage. So you just click here. Let's give 30%. So now you can see. Okay, 1, 2, 3, 4. These four are training set, and this prediction set means this test set. So we have divided the data into training set and prediction set, or you can say the test set on the basis of this one. So you can use these random selections of the sampling. Okay, on the basis of your convenient or your research methodology or your objectives, you can select this value. Okay, so as of now I am selecting 30. Okay, so other things you can leave it as it is. Of course, so many other parameters are also there. Okay, so if you are expert in QSAR model development, you can choose other way to divide the data set into training and test. Okay, so but for like basic level, this much is enough to understand. Okay, so after that you just hit click OK here. Okay, so okay, and then yes. So all previously yes, so you just click yes. Done. So we have divided the data set also uh, into training set and test sets. Now again go to analysis. Now one more option is visible in that one: variable selections and model calculations. So now we have to calculate the model. So click here. This is very important things. Okay. So combination 399 combinations, and then like mode for size 10. Okay. Significance level 0.05. Quick rules 0. Okay. Fitness. Okay. Q2 low leave, or this one. So Q2 value you can you are selecting on the basis of that one. So don't change these parameters. Okay. So but you click here GA unit, so genetic algorithms. So now here, now we have to do the hit and trial. So this is 2 means, so it will develop a models by considering the 2 variables among these 400 variables. Okay, so it will calculate, it will do the combination and permutation type of things for considering the 2 variables. Okay, and then try to develop the better models. Okay, so that is 2, and then second time you have to increase this to 3, 4, 5, 6, accordingly you have to do. And then whichever giving you the best model, you have to choose that models and then go forward. That is the way. So this is now hit and try. Okay, so now 2, 3, in that way you can do. So now very important, gene per size. So how many gene we have to run, or how many permutations we have to do. So better the gene per size, okay, better the result. So better number means generally like recommendations is above the 7,000. So but like if you are choosing 10,000, so that's better. So it will do the permutation combinations of these 2 variables for 10,000s of times. Okay, so other things you just let it be. So now this takes time. So once this uh, 2 variables is completed, you have to go for 3 variables with 10,000 gene per size, and third one is completed, you have to go 4. So how much we need them? Okay, so what is the thumb rules? Okay, so these variables number depends on your sample size. Suppose we have taken the 70, I have taken 70 compounds, right? So 70 compounds means, so each 10 compounds equivalents to one descriptors. So that means 70 compounds mean maximum 7, that one you can do iterations, or the 7 variables equations is also fine. Okay, so but lower the variables, better the equation. As per thumb rules, 10 compound can be defined by one variables. Variables mean descriptors. Okay, so 70 compounds can be defined by the 7 descriptors. That is the rules. But among the all the models, if both the models have the same parameters, suppose one model is having 7 descriptors, but all the ORCD guidelines are same, another models is having 5 descriptors, and then all the variables or the all the parameters are similar to the 7 descriptor. So you have to choose to the lower number of variables in which models is defined. That means 5 descriptors model can be better than 7 descriptor models if all other parameters are same among those 2 models. Okay, so that these are the like very important things to consider while writing or selecting the model. So now I will click. So it takes time. Okay, so it takes 10 minutes, 15 minutes to complete this thing. So you just calculate like this. So it will run in this way, like it should reach 10,000. So here you can see it is doing permutation and combination considering the 2 variables. Okay, so yeah, so it can run in the background. Is there any questions?
So while
If
it's running, I have a very small question. So while you were doing data cleaning manually, so what do you do personally? Do you completely rely on this software and clean the data, or
Mhm.
you do manually first and then you go with the software?
Yeah, generally I do manually first because I don't completely believe the software. Okay, and then like of course I take helps from the software after that, but I go first with my manual. But like you have to be very mindful while doing deleting the things, and assigning your PIC50 value. Your PIC50 value should match your structure, right? So that is also very important. So if you mismatch the PIC50 value, your whole point of developing the QSAR is wrong, right? So that's why, yeah, I do manually, and sometimes I use RD kit, uh, or these things like Python for PCA analysis and all the
Sir,
things. Yeah,
uh, while you were creating the generating these descriptors, and you downloaded that Excel file, so the sequence of the data remains same or it
yeah,
changes? Because you're saying that we have to see manually the PIC50 value and the sequence.
yeah. It remains same usually, okay, but still you have to do cross verification. So like this was the result, right? So we got it. Data remains the same, okay. Like the numbering remains the same, but the name sometimes keeps changing. So because before our name was different, right? So now AutoZane will be there, okay. So in that way, like you have to cross verify these things, but most of the time it remain the same, okay. So how you can cross verify either the compound one is this one or not? So either you can check the like these things, what is that, physiochemical properties or the molecular weight, to just cross verify for one or two compounds. If that is matching with your the raw data, previous raw data, then you can go for it. So now you can uh, type here, right? So let's say here, so here you can type now p I say 50 value, and then you cross verify this first file or any in between file. In that way you can cross verify. If that is matching, means now you can put your PIC50 value from previous Excel file to here, and then of course after that you increase all the things, whatever the criteria I told you, so you can do this manually. Yeah, but yeah, of course file name change, like AutoZone will come in paddle.
Also, when
Yes.
we were choosing the training data and test data, so can we like go first 20% we will choose as the test data, and then rest 80% we will choose as the the other part of the data.
Yeah, you can
We
do
just
that.
have to go random.
Yeah, it's random selections. Software automatically select. Okay, so every four compound will be selected as a training set, and after that one test set. So that is the way of random selections in this software. So but yeah, you can choose 20 by 80 also, there is no problem, but like that may create a problem in validation of your models. Why we are converting this 20% into test set is those 20% of the compounds variables are used to validate the data, sorry, validate the model. So that is internal validations, right? So that is dividing is on the basis of the validation, so that requires for the validation. But yeah, you can do, it's a heat and trial. Now after that, here also I am saying, right, so now it's running for two variables, right? So after this, obviously in two variables you will not get the good results, so after that you have to run for three variables, right? So you have to keep running until you are getting the best model. So it's like that. Okay, so if any other questions are there, otherwise like I will, so I will
About
conclude the last part.
yeah, sure. So just one small, can you repeat why you are choosing the two variables and why it is saying that we have to, do we have to increase the variables further? And what are the parameters that you're checking?
Sorry, I didn't understand your questions.
You chose like two variables, right? So there was a drop down option, two, three, four, five, and till 10 options were there.
Yeah, yeah.
Why did you
So no,
choose two variables?
no, like we have to do from 2, 3, 4. You have to keep increasing. Of course, you can start from 4. If you are starting from 4, also the software will start from 2. Yeah, so better you start from 2. It's like that. Software automatically start from 2. If you are selecting 4 also or 5 also, I think once I tried that one, so later I found, yeah, software is automatically trying all the variables for starting from 2. So it's better to start from 2. And why I did choose the two variables? Suppose if I am getting the best descriptor or based model within two variables, okay, so then no need to do further attributions. So that is also the point to be considered, or look at this in that way. Suppose you are getting the best models in third variables or three variables, right? So every parameters are like excellent. Every ORCD guidelines, whatever are their cutoff value are excellent means, then no need to go for fourth variables. As I already mentioned, lower the number of variables, better the model. Because like chances of randomness or chances of error will be less if you have a less number of variables. Yeah, so that's the thing. Yeah, anyone from undergrad, are you understanding, or it's like you feel complex or what? So please, you can share your thought.
साथीहरू मनमा केही छ भने खुलेर सोधौँ है, सँग सल्लाह खुलेर सोधौँ।
Yeah, so this is done for two variables, it's done. Okay, so now again go to analysis. uh, Again you can do for third variables like this. Here you can choose the 3. Okay, that is going to take time. Let me explain the analysis part. So after that you have to go to the view and select models. Okay, so view and select models, so you will be directed to this way, this one. Okay, so now the first things is, so here like status is given, right? So you mean unselected. If you click, double click it, so it will open this. If you want to select this one, so you have to click this, and here yes is there, so this is select. Now you have selected this model. So now what is this point is, so our only two variables are there, and software automatically take one variable, this one model also, right? So using these variables, software developed this 19 model. Okay, so 19 models. So if you select this one, okay, if you select this one, so now you are selecting the models. So here are the things we have to consider. So now you have to match these all the parameters with our ORCD guideline, which I explained yesterday. Okay, so on the basis of that, you have to select which one is best. Let's see R square value. So as per ORCD guideline, it should be 0.7, right? So above 0.7. So R square is 0.8. Good. Okay. So R square adjusted, I think it should be above 0.5 or 0.6. It's 0.8, right? So accordingly, these all the parameters you have to take. If all the parameters are, excellent, then you have to select this one. Okay. So before selecting, what you have to do, you have to do the internal validations and external validations, because now if you see here, okay, value for internal validations and external validations are not given in this model. Okay, so there are no parameters for the internal and external validation. So first you do this one, you click this V internal model validations. Okay, so now you validate it. Yes, so now you got new parameters over here, right? So this internal validations is also quite good, like Q square, leave more out, okay, R square, Y scrambling, this RMSE. Actually, RMSE is quite high. It should be very less. Okay, so RMSE, yeah, this is quite high. Mm, high. Yeah, so this is internal validation parameters. Okay, so let's do the external validation parameters also. So this red V is there for the external validations. You click it. So okay, so now you got more value here. Okay, so those all value are for external validation. So you have to check this one. This Q square minus F or this K value, K dash K, these two were the external validation, right? So as per our varsity guidelines, so this K dash should be above 0.85 to 1.15, and this one also the same, 0.85 to 1.15. Now here we can see, right? So our all these previous parameters are very good, but if you validate your model, okay, so your models is not quite validated, correct? So now in this way, like we have to do, so because it is showing only for 4 models, right? So because I have selected only 4 models, if you want to do remaining models, you have to select this. Okay, so in that way you can do. So now this is the things. Okay, so after that, suppose you found, let's say this model 1 is best for assume, let's assume. Okay, so to make you understand how to analyze the things, so you right click, double click here, okay, on this S, double click, so you will be directed towards the these panels. So here those all the parameters are given, which just now we explain in this column or row. Okay, so these things also you can again cross verify with our ORCD guidelines and try to match. Okay, and very important part is this one. So from these value, we are making the equation. Okay, this is intercept. Okay, and this is one variables. This is another variables. This is the coefficient for that variables. Okay, and this is the coefficient of this GATS1 as variables. Okay, so by using this one, so this is the now this is, since this is linear regression curve, linear regression models, right? So multilinear regression actually. So we are following that models equations like y is equals to mx plus c type of things. Okay, so now this is the part from where you can generate the equation. Okay, so I will show you in just a minute. Uh, Except that the plot which we can keep in our manuscripts, you can get it from here, all the plots. Okay, so you just click this. Okay, so this is in point experimental versus predicted. So this is the plot. Look at this plot. Usually the ideal, because it will look good, because our this R square value was pretty good, right? So 0.80, that's why this plot is good. Okay, so now this you can download it or save it. So you have an option here, save image, so you can save it as an image. Okay, so wherever you want to save, let's say in webinar I am saving, so in this way you can save. Okay, so similarly another one is there. So this is experimental versus predictions, but on the basis of LOO. Okay, LOO means leave out this one, what is it, like leave one out. Okay, another one will be leave all out. So these are the statistical method, okay, for the uh, relation to find out the relationship between the variables and then response. Okay, so this one also you can download accordingly. Obviously this will also look good because R square value was 0.8. These two are done. So this third one is so this is the residual calculations based on the model equations. So this calculations is based on this this model equations. Okay, so this can also be included in, and this is also residuals, endpoint versus residuals. Okay, and this one QQ plot. So this is validation plot. Okay, Q1 Q minus Q plot is, this is internal validation plot. Okay, so this plot also, actually this plot is also quite good. I think if we check our internal validation parameters, so yeah, because our internal validation parameters was also within the limit of ORCD guideline, that's why this plot is also pretty good. This is for internal validation, so this is also for the internal validations, but in another that z value versus the residue. So this is for internal validations. Okay, so this was for the R square value, and now here also Q square minus, what we did here? Yeah, so here we already did. So this one. Okay, so this is for external validations, or the, we can say, the applicability domain, AD. Okay, so this is Williams plot. This should be good, so that hat I by I as star value is 0.176. For this one there is a formula. Okay, this is the formula. From there they got the 0.176. So it calculate on the basis of number of descriptor used to design the models, and then total number of compound or the total number of training sets. So on the basis of that they calculate these things. One formula is there, you can check it by the equations or by the publications. Okay, so basically what it should be ideally. Okay, so this all the training set, because like this yellow one is the training set and this another one is the predicted set. This is the compound name. Okay, so you can choose those by the name also. So if you choose, so this is the 55 compounds. Okay, now you can check your Excel file. So what was the structure of that 45 compound, what was the structure of that this compound, or what was the structure of this 11 compound number 11. Okay, so what does it mean? So what it should be ideally, so this dash line is there, right? So this board, this board, all the values should belongs in this, within this criteria, because here 0.176, right? So here 0.176 quite here. So this is here and this is here. So within this rectangle, all the training set and then prediction set should belong. Okay, if they are going outside this, that means something not good, or the not accurate in the model. But this looks quite acceptable, because only 3 compounds are out of the uh, ideal criteria. Okay, 3 compounds are acceptables. It's not that big problem. So this is for validations. Okay, so this one also you can download it. Okay, so the same thing in Williams plot another way. Okay, and then these things is also there, like applicability domain. This is also the extended form of Williams plot. Okay, so this is very required. So this is for validations of the model. Okay, so it will also show the similar data as Q, that Williams plot. So here also one this and then one this. So these two are out of range, but it's fine. And this one, Lumo, leave multiple out. So yeah, so this is the predicted model, and red one is 2 LMO. This is model Q2. This is not good. Actually, the values should concentrate in one particular point. So these all the values should concentrate in one particular point. So this is not quite good, because like our Q square LMO value is not good here. I think if you check here that Q square LMO somewhere it will be there. So that is not good. Yeah, if you check somewhere it will be okay. So yeah, so on the basis of that one, so and another one, last one is this. We check this one, and this last one is this one. The same thing, they are presented in KXY versus the R square Y scrambling, what we called. So this will give you how much what is the chance correlations. Okay, either your model is by chance or not. So this is the Y scrambling. So validation parameters. Another very important point need to be considered if you need the PCA descriptor. It doesn't get my idea. This one, correlation parameters matrix. Okay, so here these correlation metrics should be within the range. So here 0.70, 0.80, 0.90. So this color is also there. Okay, so yellow color, I don't know what is this color. This is red color. So if you are observing any of these color here, okay, that means that model is not good. But in our case, there is no such things, right? So fine, that means like our correlations coefficients or the correlation matrix is also up to the mark. So yeah, so by considering all the parameters, the model looks good, pretty good, but external validations parameters are not up to the mark. Okay, for the external predictions also look at this. So this is 8.2, but as per the ORCD guidelines, it should be above 0.85, right? So this should be also above 0.85 to 1.15. So that is the only concerning factor in this model. Other than that, like everything looks good in this model. So in this way, now you can analyze the all other models. So if you are finding any good models, you can go with two variables. If not, again you can go for third variables and fourth variables, fifth variables. You have to keep doing these things until you are getting the base model. So that is the things. Okay, so now I will show you how to make a equation. So I told you from here you can make a equation. So let me open the Excel file. Okay, so here let's see, this one is there and this one is there. So PIC 50. So what is the PIC 50? Is equals to, it's like y square, y is equals to mx plus c type of form. Okay, so the first value, this intercept is there, right? So 12.76. So you type that one, 12.76. Okay, and after that you can type this one, this value, this second first descriptors. So along with sign you have to write. So this is minus, right? So that's why we will give here minus. Okay, and how much, what is the value? 0.0242, and then you have to give into these descriptors. What is the descriptor? ATS7V. Okay, so now this is done. So now second descriptors is there. So what is the value? Minus. Okay, so if the value is in positive, you have to write it here positive. If the value is in negative, you have to write it in negative. So this is negative 14.1415 into the descriptors. Okay, GAT, yes, 1, yes. So this is the QSAR model equation. So now by using these equations, you can predict the PIC50 value of new compound which you are going to design. Okay, suppose you are designing the new compounds, and in the same way you will minimize the structure of that new compound, and you will calculate the descriptors. From that calculated descriptor, you will take the value of these AATS7V descriptors and replace this with that original value, and then this with original value, and you will do the calculations. Whatever the value you are getting after calculations, that will be your, let's say you got 6.21. So that will be your PIC50 value. So now this PIC50 value you will convert into IC50 value, so which you are going to report in your manuscript. So with this, your QSAR is done. But analysis part is there. Okay, what does this mean here? Okay, let me say. So now how you will understand what is these parameters or what is these descriptors? Okay, so you have to find out this by this one. What is that? Previous articles, or there is one website, okay, available. I forgot the name. So that website is there. In that website, meaning of these all 1800 descriptors have been given. So But let me explain what does it mean. So like this first 12.76, that is your intermediate, okay, but after that this value is in minus. Okay, so that means this AAT as 7V descriptors is inversely related to your biological activity, and this descriptor is also inversely related to your biological activity. So if you increase the value of these descriptors, your biological activity is going to decrease. That means these types of characters from your new compound, okay, so if you have to remove these types of characters from the new compound so that you can increase the biological activity. That's what it means. If the plus sign would have been in this place, that means like these descriptors have directly relations with the biological activity. That means you want to incorporate these types of characters in your new compounds. But in this case, you don't want to incorporate these types of character in your new compound because this is inversely related to your biological activity. Yeah. So this is, I think, pretty much all about the QSAR model development, validations, and analysis. Hopefully, uh, you got some idea from here. Yeah. So if anything is there, so I can answer. We can do question answer round. Otherwise, like from my sides, like QSR is pretty much clear in all the aspect.
So can I go on?
Yeah, yeah, please.
So when you choose two variables and you ran the sample, so let's assume we got some parameters out of mark. So that time, do we have to repeat the whole process and run the same samples with three variables, or maybe I don't know. So in that case, what do we do?
What? But I didn't understand your question. Can you repeat, please?
Suppose when you chose in that application, you chose two variables, and there were few parameters which were not up to the mark, like k values.
Yeah.
So if we are not getting good results, so do we have to repeat the whole procedure in that application with different number of variables?
Yeah, not the whole process. Not whole process. Here only you have to repeat the things. Okay, let me accept this. Here only, like analysis, you have to go variable selection mode. So now you have to go for third variable. So it will automatically calculate. Okay. So or you can choose here four variables, or it will automatically calculate. So that's what you have to repeat. Not the whole process. From starting, no need to do. Okay. So if you are, trying all the variables, but still you are not getting the accurate models, then in that case, you have to do from starting. Okay. So yeah, till then you have to keep trying. So since I have taken the 70 compounds, so I can try till 7 variables. If I am not getting the best models till 7 variables, then something is wrong in your data cleaning, or data splitting in that one. In that case, you have to go back from scratch and start. Otherwise, variable selections you can do from here. No need to go previous one.
Also, one thing that while we are like publishing any article regarding the QSAR, so how much data we have to give them? Do we have to provide
All
this
the
data
data.
to?
All the data. Yes, of course. Like this raw data also we have to provide in electronic form, or yesterday I told you one website free. Either in GitHub you have to create your ID and upload all the data and make a that one, what is that link, and provide that link, or some other repository sites are there. There you have to upload your raw data I am talking about. Okay. Of course, this plot and all the things you will upload in the manuscript, so no need to keep those plot. But the raw data, let's say the data selections, these Excel files, PIC50, how you selected, what how many compounds you selected initially. So these are all the informations you have to provide to the journal, that is best practice. uh, in repository form. So in repository, you have to upload and provide them the link. Not only for these things, QSAR, okay? So while publications for all other type of publications also, it is good practice if you are providing them the data. From docking also, MD simulations or network pharmacology, whatever you are doing, it's always good to provide the raw data because that makes your research authentic. Yeah, that's it from my side. If any other questions are there, we can address. Otherwise, I think, yeah. Please over to Dr. Dirgha sir.
Thank you very much, Shankar sir, and all the participants for your time in our mercy for quite meaningful discussion. I hope definitely it will help us to grow more in QSR hence on coming days. साथीहरू तपाईँहरू कसैको मनमा केही शङ्कर सरलाई भन्नुपर्ने छ, पोजिटिभ सुझाव छ भने पनि दिन सक्नुहुन्छ। र यो फोरमलाई युज गरेर हामी यो रिलेटेड रिसर्च माइन्डसेटहरूले एउटा नेटवर्क जस्तो बनाउँदै जाने। तपाईँहरू कोही कनेक्ट हुनुभएको छैन भने, if you are not connected with the Association of Faculties of Pharmacy of Nepal network, you can follow the Facebook page and get update from there. We used to host similar kind of webinar. We plan to do some more webinar in coming days in different topic. If you can suggest some new topic, we can try that also. We will find the resource person, and our intention is to make the resource available to everyone, simplified more as possible, so we can grow together. And if anyone have any question, please feel free to ask. Be very much specific and quick.
Yesterday, I was asking if sir is ready to collaborate so that sir can share his contact.
That we can discuss later after this meeting. You can share the proposal, how we can collaborate and work together. Definitely we can find that option, yeah.
I have shared my email ID in the chat box, so you can use that one for other things. Yeah. And then I want to suggest one thing. Okay, if you are using all the software which I explained yesterday and today, please don't forget, if you are publishing the data by using this software, please don't forget to cite their relevant articles. Okay, so I will you just try to find out the relevant articles regarding the software. Okay, so like QSAR has three articles under the same software, so you have to cite those software for the recognitions of contributions for their models. So that is very important.
Then okay, yeah, if you do not have any more question, we can wrap up today's webinar as well. आज हामी झन्डै 25जना जति सहभागी थियौँ, हिजो अलि बढी थियौँ, अहिले अब सकिन लाग्दा धेरै छुट्टिसक्नु भएको छ। आगामी दिनमा पनि हामी फेरि भेट्नेछौँ र सम्भवतः यो 2 दिनको वेबिनार म हाम्रो आइटी टिमलाई भन्दैछु, उहाँले टाइम म्यानेज गर्न सक्नुभयो भने very soon we will make this episode available in our AFP and YouTube channel soon, and you can follow that again and practice yourself. यदि केही समस्या भयो भने फेरि शङ्कर सरसँग कनेक्ट गर्ने, मसँग कनेक्ट गर्ने, हामीसँगै छौँ है त सिकाइमा। हस् शङ्कर सर, हजुरको समयका लागि अत्यन्तै धेरै आभारी छु, जुन यो लामो समय झन्डै 4 घण्टाको, हिजो 2 घण्टा आज 2 घण्टा बढीको समय दिनुभयो, त्यसका लागि अत्यन्तै म आभारी छु। धेरै धन्यवाद र हजुरको ग्रेजुएट लाइफका लागि धेरै शुभकामना। मलाई सबै विद्यार्थीहरूलाई पनि त्यही सुझाव छ कि शङ्करजी जस्तै हामीले मेहनत गर्न सक्नुपर्छ। हामीले पिआईलाई मात्र कुरेर हुँदैन, आफ्नो गुरु आफै बन्नुपर्छ जीवनमा कतिपय क्षणमा, र अत्यन्तै धेरै मेहनत गर्ने, र त्यो मेहनत गर्ने क्रममा शिष्य यदि हङ्गरफुल छ भने गुरु पक्का भेट्छ, उसले सहजै भेट्छ। त्यसैले जसले जानेको मान्छे हुन्छ ऊ नलेज सेयर गर्न चाहन्छ, तर त्यति हो कि हाउ मच हङ्गर डु आई होल्ड, होइन, त्यो हङ्गर चाहिँ क्रिएट गर्न मैले सक्नुपर्छ। त्यति भन्दै शङ्कर सरको मनमा केही छ भने उहाँले भन्नुहुन्छ, होइन भने हामी छुट्टिन्छौँ। शङ्कर सर केही छ क्विकली?
Fine, sir. Like, thank you for all the participations, and then I think it was. I hope it was fruitful conversations and then webinar. I only want to suggest all the young researchers and then who are eagerly willing to do the CAD research. So please follow or do the experiment using authentic source, authentic software. So whatever the software we practice yesterday and today, so you can use those software. And please don't forget to give the contributions of their software and cite their articles. I'm not saying their individual articles. I am saying the articles which is related to this particular software. So that we have to do. And then most importantly, thank you for the participations. अनि के थियो, म अर्जुन सर हुनुहुन्छ होइन सर, आज कि जानुभयो? अर्जुन सरको
अर्जुन सर यहीँ हुनुहुन्छ।
सरले मलाई कन्ट्याक्ट दिनु होला, म दीर्घ सरसँगबाट पनि लान्छु। आई थिङ्क आई वान्ट टु डिस्कस अबाउट म आई थिङ्क के भन्छ सर, केही डिस्कस गर्छु म अर्जुन सरसँग। या द्याट्स इट।
ए हुन्छ हुन्छ। अर्जुन सरको बढी यो एप्लिकेसन डेभलपमेन्ट अनि एआई, मेसिन लर्निङ अन्य कुरामा उहाँको स्किललाई हामीले युज गर्न सक्छौँ। जस्ट उहाँलाई चाहिँ अलिकति यो हेल्थ रिलेटेड माइन्डसेट चाहिँ हामीले इनपुट गर्न सक्यौँ भने हामीले ब्युटिफुल्ली केही काम गर्न सक्छौँ शङ्कर सर।
म त्यही सोचिरहेको थिएँ सर, त्यही भएर उहाँसँग डिस्कस गर्नुपर्छ।
हुन्छ हुन्छ, म कनेक्ट गराउँछु नि सर है। हस् हस्,
ल
ल ल, शुभरात्रि। थ्याङ्क यू एभ्री वान, गुड नाइट।